Hello everyone. Now we're going to be covering Campbell Biology Chapter 13, Meiosis and Sexual Life Cycles. Here, when it comes to one of our beginning figures, what accounts for the resemblance between offspring to their parents? Well, that's because of the sexual reproductive process called meiosis, which produces cells with half the chromosomes of each parent, and it occurs only in specialized cells. Meiosis will occur both in the father and in the mother, specifically in the father's testes and in the mother's ovaries, starting with a parent cell. In the case of humans, we have 46 chromosomes. Only one pair of homologous chromosomes, or homologs, is shown here. Out of those 46, those chromosomes are duplicated before meiosis begins. During Meiosis I, homologs are separated so that you have two daughter cells. And then during Meiosis II, sister chromatids are separated as well, so that starting parent cell eventually leads to 4 cells, which in the case of the father, that'd be 4 sperm cells. Each chromosome is one of a pair of homologs, whereas in the mother, those 4 cells would become egg cells, though oftentimes one of those cells might inherit more of the material for the cytoplasm, while the other 3 might not be functional. Fertilization is when one sperm cell with half the genome necessary for that species and an egg cell with half the genome of that species come together to make the full genome for that species. In the case of humans, where half of the chromosomes come from the sperm and half of the 46 set of chromosomes come from the egg, and then you end up with 46 total chromosomes after fertilization. Concept 13.1. Offspring acquire genes from parents by inheriting chromosomes. The transmission of traits from one generation to the next is called inheritance or heredity. Sons and daughters are not identical copies of either parent or of their siblings. Along with inherited similarity, there is variation. The study of heredity and inherited variation is called genetics. Inheritance of genes. Genes are the units of heredity and are made up of segments of DNA. Genes are passed to the next generation via reproductive cells called gametes, which could be the sperm and the eggs. Most DNA is packaged into chromosomes. Humans have 46 chromosomes in the nuclei of their somatic cells. All cells of the body except gametes and their precursors would be somatic cells. A gene's specific position along a chromosome is called a locus. Comparison of asexual and sexual reproduction. In asexual reproduction, a single individual passes all of its genes to its offspring without the fusion of gametes. A clone is an individual or group of genetically identical individuals from the same parent. In sexual reproduction, 2 parents give rise to offspring that have unique combinations of genes inherited from the 2 parents. On the left here, we have a hydra, which can reproduce. One example of reproduction through budding, leading to a complete an offspring that is identical to the parent. And redwoods we have on the right. Asexual reproduction of hydra through budding. If you have your e-textbook, you can see this video. Concept 13.2. Fertilization of meiosis. Alternate in fertilization and meiosis alternate in sexual life cycles. A life cycle is the generation-to-generation sequence of stages in the reproductive history of an organism. The behavior of chromosomes is related to the human life cycle and other types of sexual life cycles. Sets of chromosomes in human cells. Human somatic cells have 23 pairs of chromosomes. A karyotype is an ordered display of the pairs of chromosomes from a cell. The two chromosomes in each pair are called homologous chromosomes, or homologs. Chromosomes in a homologous pair have the same length, centromere position, and staining pattern. They also carry genes controlling the same inherited characters. Here we have bioinformatics showing some applications. About analyzing genetic material. In this technique, we have a pair of homologous chromosomes. The metaphase chromosome is duplicated, and we have sister chromatids. The centromeres, one per sister. For centromeres, there's one per sister chromatid. And then we have our karyotypes. For humans, chromosomes 1 through 23, or in this case, what's being shown is the non-sexual chromosomes 1 through 22. The sex chromosomes, which determine the sex of the individual, are called X and Y. Human females have a homologous pair of X chromosomes. Human males have a single X chromosome and a single Y chromosome. The remaining 22 pairs of chromosomes are called autosomes, which are non-sex-defining chromosomes. At this point, as always, if you feel inclined, you can like the video and subscribe to the channel, or of course leave a comment, but none of that is necessary, but just fun to see. Each pair of homologous chromosomes includes one chromosome from each parent. The 46 chromosomes in a human somatic cell are two sets of 23, one set from the mother and one set from the father. A diploid cell, represented by the shorthand 2N, has two sets of chromosomes. For humans, the diploid number is 46, or 2N equals 46, and being the number of sets, 23. Or number of pairs. In a cell in which DNA synthesis has occurred, each chromosome is replicated. Each replicated chromosome consists of two identical sister chromatids. A gamete, which would be either the sperm or the egg cell, contains a single set of chromosomes, and is thus referred to as a haploid cell, or with the shorthand N. For humans, the haploid number is 23. Each set of 23 consists of 22 autosomes and a single sex chromosome. In an unfertilized egg or ovum, the sex chromosome is X. In a sperm cell, the sex chromosome may be either X or Y. For this figure, the key here is for those of you who can read in color, we have red being the maternal set of chromosomes and blue being the paternal set, or in other words, red being where my cursor is right now. And now for the paternal set, where my cursor is highlighting at this point. Sister chromatids being shown, pairs of sister chromatids of one single duplicated chromosome. Each sister chromatid has a centromere. And toward the bottom, we see two non-sister chromatids in a homologous pair, or a pair of homologous chromosomes, one from each set, one from the maternal, one from the paternal. Behavior of chromosome sets in human life cycle. Fertilization is the union of gametes, the sperm and the egg combining together. The fertilized egg is called a zygote and has one set of chromosomes, one from each parent. The zygote produces somatic cells by mitosis and develops into an adult. So here we have another figure where the green arrows, the top half going from meiosis to fertilization, represents the haploid period, where haploid represented by the number N. For humans, that would be 23. And then the bottom half, represented by orange arrows from fertilization, that is the diploid point, leading to fertilization, and then mitosis and development leading to a full organism. Also, a point where we have a diploid number is the initial part of our parent cell, which has the full genome of either the paternal end or the maternal end, but then through meiosis, they eventually divide into haploid cells. The ovaries and testes produce haploid gametes. Gametes are the only type of human cells produced by meiosis, rather than by mitosis. Meiosis results in one set of chromosomes in each gamete. Fertilization and meiosis alternate in sexual life cycles to maintain chromosome number. The variety of sexual life cycles. The alternation of meiosis and fertilization is common to all organisms that reproduce sexually. The three main types of sexual life cycles differ in the timing of meiosis and fertilization. As shown on the left, we have what's common to animals. We have a diploid cell that undergoes meiosis and becomes a set of haploid cells that then become fertilized from another haploid cell to form a diploid. And then when that organism grows into adulthood, it can then continue the meiosis process to produce other gametes. In the middle, we have examples of what's seen in plants and some algae, where we have meiosis occurring, and then those gametes grow larger through mitosis, multiply, and then continue on through mitosis, and then are fertilized by the combination of haploid cells forming the diploid cell. What we see here is some plants might be haploid multicellular organisms, such as a gametophyte, and then they come together to form a diploid multicellular organism called a sporophyte. And then on the right, we have what is shown in most fungi and some protists. Haploid unicellular or multicellular organisms are possible, where we initially have our diploid organism or diploid cell that undergoes meiosis, then different series of mitosis, eventually leading to fertilization of haploid cells that again make the diploid. Gametes are the only haploid cells in most animals. They are produced by meiosis and undergo no further cell division before fertilization. Gametes fuse to form a diploid zygote that divides by mitosis to develop into a multicellular organism. As shown with animals, we have our diploid cell. It grows, it multiplies during mitosis, but then when it's time to start taking those sex-specific cells, those gametes are formed by a diploid cell undergoing mitosis, and then two haploid cells, one from each parent, coming together to fertilize and become a diploid cell that eventually multiplies through mitosis to form a multicellular organism. Plants and some algae exhibit an alternation of generations. This life cycle includes both a diploid and haploid multicellular stage. The diploid organism, called the sporophyte, makes haploid spores by meiosis. Each spore grows by mitosis into a haploid organism called a gametophyte. Gametophyte makes haploid gametes by mitosis. Fertilization of gametes results in a diploid sporophyte. So essentially, the diploid multicellular plant can undergo meiosis to form gametes, whereas the haploid multicellular organism, the gametophyte, simply goes through mitosis until fertilization occurs between two haploid plants, to form the diploid offspring. In most fungi and some protists, the only diploid stage is the single-celled zygote. There is no multicellular diploid stage. The zygote produces haploid cells by meiosis. Each haploid cell grows by mitosis into a haploid multicellular organism. The haploid adult produces gametes by mitosis, as seen in this graphic here. Depending on the type of life cycle, either haploid or diploid cells can divide by mitosis. However, only diploid cells can undergo meiosis. In all three life cycles, the halving and doubling of chromosomes contribute to genetic variation in offspring. Concept 13.3. Meiosis reduces the number of chromosome sets from diploid to haploid. Like mitosis, meiosis is preceded by the replication of chromosomes. Meiosis takes place in two consecutive cell divisions, called Meiosis I and Meiosis II. The two cell divisions result in 4 daughter cells, rather than 2 daughter cells in mitosis. Or the 2 cell divisions of meiosis result in one parent dividing into 4 daughter cells, while in mitosis the parent cell only breaks up into 2 equivalent daughter cells. Each daughter cell has only half as many chromosomes as the parent cell after Meiosis II is finished. Here we see interphase, where a pair of homologous chromosomes in the diploid parent cell. Those chromosomes are duplicated, and a pair of duplicated homologous chromosomes come together as sister chromatids, each homologous chromosomes consisting of sister chromatids. The diploid cell with duplicated chromosomes then undergoes meiosis I, where those homologous chromosomes are separated. The haploid cells with duplicated chromosomes then each have a set of chromosomes, and then each of those cells undergo meiosis II, where sister chromatids are then separated, forming haploid cells with unduplicated chromosomes. And here's how we go from one parent cell to 4 daughter cells, thanks to meiosis. The stages of meiosis: chromosomes duplicate before meiosis. The resulting sister chromatids are closely associated along their lengths. This is called sister chromatid cohesion. The chromatids are sorted into 4 haploid daughter cells. Divisions in meiosis I occurs in 4 phases, which are prophase I, metaphase I, anaphase I, and then telophase I, along with cytokinesis. For prophase I, in early prophase I, each chromosome pairs with its homologue, and crossing over can occur. X-shaped regions called chiasmata are sites of crossovers. And essentially what that means is there's a possibility of genes from each switching over to the other, helping cause variability. Metaphase 1, pairs of homologs line up at the metaphase plate with one chromosome facing each pole. Microtubules from one pole are attached to the kinetochore of one chromosome of each pair. Microtubules from the other pole are attached to the kinetochore of the other chromosome. During anaphase 1, pairs of homologous chromosomes separate. One chromosome of each pair moves toward opposite poles, guided by the spindle apparatus. Sister chromatids remain attached at the centromere and move as one whole unit toward the pole. In telophase 1, in the beginning, each half of the cell has a haploid set of duplicated chromosomes. Each chromosome still consists of two sister chromatids. Cytokinesis usually occurs simultaneously, forming two haploid daughter cells. In animal cells, a cleavage furrow forms. In plant cells, a cell plate forms. No chromosome replication occurs between the end of Meiosis I and the beginning of Meiosis II, because the chromosomes are already replicated. So what we have here is the entire sequence of Meiosis I, which separates homologous chromosomes. And you can pause the video if you want to look at this further in detail. Divisions of Meiosis II also occurs in 4 stages: prophase, metaphase, anaphase, and telophase II, along with another set of cytokinesis. Meiosis II is actually very similar to mitosis. Prophase II, a spindle apparatus forms. In late prophase II, chromosomes, each still composed of 2 chromatids, move toward the metaphase plate. In metaphase II, the sister chromatids are arranged at the metaphase plate. Because of crossing over in Meiosis I, the 2 sister chromatids of each chromosome are no longer genetically identical. The kinetochores of sister chromatids attach to the microtubules extending from opposite poles. In anaphase II, the sister chromatids separate. The sister chromatids of each chromosome now move as 2 newly identical chromosomes toward opposite poles. In telophase II, the chromosomes arrive at opposite poles. The nuclei form and the chromosomes begin decondensing. Cytokinesis separates the cytoplasm. At the end of meiosis, there are 4 daughter cells, each with a haploid set of unreplicated chromosomes. Each daughter cell is genetically distinct from the other and also from the parent. And here we have a graph showing Meiosis II from prophase, metaphase, anaphase, and telophase II, and the cytokinesis stage in Meiosis II. Crossing over, and a process I like in particular, synapsis. During prophase I, after interphase, sister chromatids are held close together by proteins called cohesins. The non-sister chromatids are broken at precisely matching points. The zipper-like structure called the synaptonemal complex holds the homologs together tightly. During synapsis, DNA breaks are replicated, joining DNA from one non-sister chromatid to the corresponding segment of another. And here we have a chart displaying that. We're in part 1. On the top left, we have DNA breaking, cohesions binding, the centromere still being the point where those sister chromatids are together, And then DNA breaks as well. There's one set that is the paternal sister chromatids and one set of sister chromatids that are maternal. During stage 2, the synaptonemal complex begins forming. In part 3, sister chromatids as shown. And then we have our synaptonemal complex. And then there is chiasmata, or a crossing over, where those non-sister sets of chromatids are bound tightly to each other. A comparison of mitosis and meiosis. Well, before we continue further, during this synapsis process, at the chiasmata, it's possible for genes to actually swap places. So a gene that might only be from the paternal set of sister chromatids might be replaced with a similar gene from the set of sister chromatids. While this doesn't always happen, it is possible for genes to swap, and that helps with having some pretty awesome variability. Now, though, a comparison of mitosis and meiosis. Mitosis conserves the number of chromosome sets, producing two cells that are genetically identical to the parent cell. Meiosis reduces the number of chromosome sets from diploid or 2 to haploid or 1, producing 4 cells that differ genetically from each other and from the parent. 3 events are unique to meiosis, and all 3 occurred during Meiosis I. First is synapsis and crossing over. During prophase I, homologous chromosomes physically connect and exchange genetic information. 2, there is alignment of homologous pairs at the metaphase plate. And 3, there is separation of homologs during anaphase I. And here we're just seeing a comparison of mitosis and meiosis. And if you like, you're more welcome to pause the video to get a good look at this stuff. Sister chromatid cohesion allows sister chromatids to stay together through Meiosis I. In mitosis, cohesions are cleaved at the end of metaphase. In meiosis, cohesions are cleaved along the chromosome arms in anaphase I, the separation of homologs, and at the centromeres in anaphase II, the separation of sister chromatids. Concept 13.4, genetic variation, produced in sexual life cycles, contributes to evolution. Mutations, such as changes in an organism's DNA, are the original source of genetic diversity. Mutations create different versions of genes called alleles. Reshuffling all alleles during sexual reproduction produces genetic variation. Origins of genetic variation among offspring. The behavior of chromosomes during meiosis and fertilization is responsible for most of the variation that arrives in each generation. 3 mechanisms contribute to genetic variation: independent assortment of chromosomes, crossing over during synapsis, and then random fertilization. What sperm cell meets which egg cell? Independent assortment of chromosomes. Homologous pairs of chromosomes orient randomly at metaphase I of Meiosis I, or at metaphase I of meiosis. In independent assortment, each pair of chromosomes sorts maternal and paternal homologs into daughter cells, independently of the other pairs. The number of combinations responsible are possible when chromosomes assort independently into gametes is 2 raised to the nth power, where n is the haploid number. Humans, that's 23. Therefore, there are more than 8 million possible combinations of chromosomes that can occur to form a sperm or an egg. And then the combination. So, for example, during one set of meiosis, one parent cell, the following are two equally possible arrangements of chromosomes at metaphase 1, where all the maternal is at one end and all the paternal are at one end. Then they break up, and you're left with two cells that just have maternal DNA and just have paternal DNA. And then each leads to a combination where there are two daughter cells that just have maternal DNA and just have paternal DNA, though that's very unlikely. Another illustration is you have, during that random assortment, some of the paternal sister chromatids are on one side and the other are on the other side. Some of the paternal sister chromatids are on one side and some align on the other. Therefore, during metaphase 2, you have a combination of DNA from both parents of that person, of that individual, and therefore the daughter cells, either the egg cells or the sperm cells produced by that person, can be a combination of that person's father and that person's mother. So there's a huge range of possible combinations thereof. During crossing over produces recombinant chromosomes, which combine DNA inherited from each parent. Crossing over contributes to genetic variation by combining DNA from two parents into a single chromosome. In humans, an average of 1 to 3 crossover events occur per chromosome, which is amazing for how much DNA is involved. Here what we see is, in prophase 1 of meiosis, non-sister chromatids, so a set from the father and a set from the mother, are held together during synapsis. And therefore we have a pair of homologs. Synapsis and crossing over occurs. At the chiasmata, that's where these are tightly bound and pinched, and we have the centromeres that are holding the sister chromatids together, but the chiasmata that is holding the non-sister homologs together. Movement to metaphase 1 plate occurs, and then as seen in anaphase 1, there's a breakdown of protein holding the sister chromatid arms together. However, due to the binding of the chiasmata, we see here that there was some crossing over, where, for anyone who's colorblind, this side was just red and this side was just blue, but now the blue side has a little bit of red that crossed over, and the red side has a little bit of blue that crossed over, thus leading into a recombinant form of sister chromatids that eventually become split into chromosomes, some of those chromosomes might be from one specific parent of that person, and some might be a combination of that person's parents. And this leads to the formation of the egg cells or the sperm cells that, once they fertilize each other, can lead to a new combination, unique from that person's parents and their grandparents. Random fertilization. Random fertilization adds genetic variation because any sperm can fuse with any ovum or unfertilized egg. The fusion of two gametes, each with 8.4 million possible chromosome combinations from independent assortment, produces a zygote with any of about 70 trillion diploid combinations. So, you are essentially 1 in 70 trillion of a combination of your parents. Crossing over adds even more variation. Each zygote has a unique genetic identity. The evolutionary significance of genetic variation within populations. Natural selection results in the accumulation of genetic variation favored by the environment. Essentially, an organism doesn't necessarily choose what genes it picks up, but perhaps there are certain factors in the environment that organisms with certain traits are more likely to live long enough to produce offspring, and therefore their genes are more likely have a higher probability to continue. But there are also mutations, which are the original source of different alleles, such as whether or not you have a blue eye or a brown eye or green eye in humans, or hazel. These are mixed and matched during meiosis. Sexual reproduction is almost universal among animals. Asexually reproducing organisms like the Bdelloid rotifer increase genetic diversity by incorporating foreign DNA from the environment. And bacteria are also able to just often share DNA between neighboring bacteria as well, because they don't have their DNA protected in a nucleus. And then we just have some final figures for the end of our chapter. Again, prophase 1, each pair of homologous chromosomes undergoes synapsis and crossing over between non-sister chromatids with the subsequent appearance of chiasmata. In metaphase 1, chromosomes light up as homologous pairs at the imaginary metaphase plate, and then in anaphase 1, homologs separate from each other. Sister chromatids remain joined at the centromere, as shown here. And we just have plant variation. Basically, some plants that might be more acclimated, some offspring that might be more acclimated to the weather, some that might not, some that might be more acclimated to certain diseases or aging, some that might not, and it leads to a wide range of variability. As always, if you make it to the end of the video, you're always welcome to like the video or subscribe to the channel. Neither are necessary, but they're fun to see. And as always, please consider leaving a comment just to let me know if there's any other material you'd like to hear about in our small channel, and let me know how your classes are going and wherever they are. No matter what, though, I wish you all a great semester, and peace.